Okay, so when we don't always want DNA because the DNA is actually inside the nucleus. And so how the DNA actually controls the body is that it produces proteins that basically go and do its work for him, or polypeptides, proteins, polypeptides. And so the way that the DNA does this, despite being in the nucleus, is that it has to send messages outside of the nucleus to the ribosomes, which actually produce the proteins. And so in order to send messages to the ribosome, it has to travel outside the nucleus, and the way it does that is that it forms messenger RNA, mRNA. And so messenger RNA, the process of forming that is called transcription. And so what's unique about RNA is that it actually has different bases from DNA. So while DNA has adenine, thymine, cytosine, guanine, RNA doesn't actually have thymine. Instead of thymine, it has uracil. And so we'll be representing uracil with these little triangles. So it works a lot like replication. So the DNA molecule is split up. And so another enzyme is called RNA polymerase, and what RNA polymerase basically does is that it finds free-floating bases, and it just allows those bases to match up with the various. It lets the bases match up here with all the different. Oops, sorry. So it just sticks on this long, it just follows the chain, and it just adds all the different bases. And so what's sort of unique about RNA is that unlike DNA is it only has one strand. So the final product is actually just one very long strand that is the complementary pair of each of these bases here. And so this is called the antisense strand, and this is called the sense strand. And the reason that it's named like that is basically the long RNA strand that's produced from this strand is identical to the sense strand here, except instead of thymine it has uracil. So it's basically just a copy of this strand here. It's just one long copy of this strand, only, yeah, the bases are slightly different. So this is the antisense strand, and it's the single RNA strand is basically the opposite, sort of this long antisense strand that it's actually produced from. Pause it. Okay, so this is our DNA model here. We've represented the DNA backbone like this. So all these little metal balls here are the pentose sugars that make up the backbone of the DNA. These loops here, those are the phosphate groups attached to the sugars, and since they're stuck to each sugar, it represents the covalent bonds between the sugar backbones and the phosphate groups. So they're covalent bonds sticking them together. And so our DNA gets its pattern because of something called complementary base pairing, which basically means that out of the 4 bases in DNA, each one has a complement, like there's a complementary pairing, so there's a partner for each. So we have red, blue, green, and yellow, but in real life these bases are adenine, thymine, cytosine, and guanine. And so adenine only ever attaches to thymine, and cytosine only ever attaches to guanine. And the reason this is that in our model, while they're stuck together by magnets, in real life, these middle bases here, these nitrogenous bases, are actually stuck together by hydrogen bonds. And so the amount of hydrogen bonds in each pair is actually kind of different. So in adenine and thymine, there are 2 hydrogen bonds that link them. And in cytosine and guanine, they're linked by 3 hydrogen bases, hydrogen bonds actually. So it's not actually possible for adenine to bond with cytosine the same way that it bonds with thymine as a result, because their shape is sort of different and the way they attach to their pairs is different. And so the process of replication is where the DNA molecule is turned into two DNA molecules. And the way this occurs is there is a enzyme called helicase, helicase. And so what helicase does is that it goes along the length of the molecule and it basically just breaks the hydrogen bonds that stick the nitrogenous bases together. And so DNA has two strands basically. So what happens is that when you break it apart, another enzyme called DNA polymerase comes along, and what DNA polymerase does is that it actually allows free-floating nucleotides, so DNA monomers, to attach to the new bases that have just been split up from the other strand. So DNA polymerase allows strands to just stick together to new to the various nucleotides that are floating around. And so since it's complementary base pairing, you will actually end up with two identical sets of DNA strands. So like these two here are identical because there's only one way that they can be put back together. And so it continues all the way down the line until we're left with two different molecules of DNA. But what's interesting is that if you take away all the new stuff that's been added, you can see that basically each new strand contains one half that's from the original parent, from the original strand of DNA, and one strand that's formed from the free-floating nucleotides, which we've just taken away here. And so this is why replication is called semi-conservative, because it conserves, it always conserves the original parent molecule, because each new strand, half of that is the parent molecule, which is the original strand that stuck the whole thing that the new strand is being created by. So the new strand is the one formed by the free-floating nucleotides, and the parent strand is the one that comes from the original one. So yeah, that's okay.
Okay. The mRNA locates a ribosome and aligns with it. The two codons are within the boundaries of the ribosome. Okay, so a codon is three sets of nitrogen bases. 1, 2, 3. 1, 2, 3. Then a tRNA molecule comes in. This is going to be our tRNA molecule, and this shall be our amino acid. Okay. The anticodon of this molecule must be complementary to the bases of the mRNA.
Whoa.
Then a second tRNA molecule comes in and aligns with the second one, containing its own amino acid. Oh no.
Wait.
The second anticodon is also complementary to the second codon. Okay, pause the video. Okay, and a peptide bond is formed between the two amino acids. This shall be our peptide bond. And then the bond between the first tRNA and the amino acid is broken. And fourth with the second amino acid. This is what we call a polypeptide. And then an enzyme breaks apart these tRNA from the mRNA. Oh no. And they can be used for other reactions and such. Translation.